Welcome to this edition of the Perspectives in Inflammation podcast, a series brought to you by the CSF. It shines a spotlight onto experts in the field of inflammation and their research interests. My name is Paul Emery from the University of Leeds, UK, and joining me today is my good friend, Professor Michael Weinblatt for the Brigham and Women's Hospital at Harvard University in the US. Welcome, Michael.
Thank you, Paul.
Michael, you've been at the Brigham for 40 years. Can you tell me a little bit about the institution and the clinical research programs you have there?
So our division evolved from the Robert Brigham Hospital, which was the first arthritis hospital in the United States, which was founded in 1914. That was a 96-bed hospital for orthopedics and rheumatology. And then in 1980, we merged with the Peter Bent Brigham to form the Brigham Women's Hospital. We have a huge rheumatology division with over 60 rheumatologists. We have a very large basic research group with a variety of interests. Our initial chair was K. Frank Austin, one of the world's experts in inflammation, followed by Mike Brenner, and our current chief now is Ellen Gravelisi. We also have a very large clinical research program directed by Dan Solomon and Karen Kostenbader, primarily interested in health outcomes and epidemiology, a variety of diseases. And I've directed a clinical therapeutic unit and now oversee a large patient registry called BRASS. So it's been a phenomenal place. We have a huge clinical faculty. We see over 40,000 outpatients a year. We have over 4,500 patients with rheumatoid arthritis that we manage.
So how did you get into rheumatology in the first place? You were a resident, I think, down in Maryland, is that right?
Yeah. So like many of us, it was mentoring. I had two remarkable mentors in Baltimore, Gene Jackson, who was chief of rheumatology in Maryland. And then I spent quite a bit of time at Johns Hopkins with Mary Betty Stevens, who was the chief of the program, who was really a remarkable clinical investigator and clinician. And they were my two early mentors. And then I went to Boston for my fellowship, where I had the privilege of working with K. Frank Austin and Ron Anderson, a superb clinician, who really taught me the purpose of seeing patients and what we were supposed to be doing and the value of good clinical research.
Well, I think we share the same opinion that the best questions come from seeing clinical issues and actually directing your research to where they're needed. You were there in the pre-biologics era, obviously, and there were a large number of patients to be treated. What were your approaches in those days?
Well, I think just like what you did in the UK, we used the, quote, slow-acting anti-rheumatic therapy, which in retrospect was ridiculous. Gold salts were a major part of our therapy. At the Robert Brigham, our patients were admitted to the hospital for 2 to 4 weeks, where they were put in Hubbard tanks and given hot wax therapy and injected their joints. And we started them on gold treatment and low-dose steroids. And they did well in the short term. And then we sent them home, and then they came back 3 months later to start another realm. And it was very disappointing in patients in wheelchairs, as you mentioned, ambulances. I mean, the reason our patients stayed functional back then was because of the skill of the orthopedic surgeons. We had a combined unit with orthopedics, so we all met every day. We had really skilled surgeons led by Clem Sledge, and that led our patients to be functional. But clearly, from 1978 to 1980, when I trained, we were really stalled as far as new therapies for the treatment of this disease.
Well, Michael, the story of methotrexate is an incredible one, and I'm sure everyone would like to hear your take on it. You were right there in the middle of it all, so
yeah,
can you give us a little bit about that?
So it's not like we discovered a new drug. Methotrexate had been around since its parent compound since 1948, aminopterin, in the treatment of childhood leukemia. And the observation that antifolates could work in rheumatoid arthritis were made in 1950, but the observation was lost by the rheumatology community. I think great credit needs to go to Rex Hoffmeister, practicing rheumatologist in Spokane, Washington, who reported in 1972 his personal use of methotrexate and the success, but he never published it because he had incredible criticism about using a cancer drug to treat RA. I had used methotrexate early in my career when I was at Wake Forest University, and it had been used by dermatologists for decades. And I wrote a clinical protocol. In fact, it was the first clinical therapeutic study I'd ever done. I wrote a protocol and I sent it to Letterly Laboratories. And they initially wrote back that they weren't interested. And several months later, they invited me to a meeting where John Ward, one of the real giants of clinical therapeutics, was in attendance. And Joel Kremer, a young rheumatologist from Albany who was interested in studying methotrexate. And the medical director at the time, Harriet Kilty, a Scottish lady, fantastic, said, "I'm interested in methotrexate and I'm going to fund your studies." Joel was interested in looking at the impact of methotrexate on the liver. And I was interested to find out whether methotrexate worked by designing a placebo-controlled trial. The remarkable thing is that Letterly, particularly Dr. Kilty, had the vision not just to support our initial randomized trial, but when it was published, then to allow us to follow these patients long-term. And then she gave me the opportunity to design a number of trials comparing methotrexate to other drugs. So we took an old drug and repurposed it for rheumatoid arthritis. And I think it, as most people do, is beginning of the new therapies for this disease.
Do you remember, Michael, we had a meeting where sulfasalazine was promoted as European methotrexate?
That's right. And that might be one of the first meetings where I met you, Paul.
Yeah.
It didn't hurt that it was in Italy. And we had Hilary Capel, the queen of sulfasalazine, and yourself.
4 grams a day, yes.
And we sat around and talked about drug development and where we should be going. And, if you look back over 30 years, there have been, some seminal points in the development of treatment for the disease, methotrexate being one, and obviously the development of biologics, particularly anti-TNF therapies, really changed this disease.
Yeah, Mike, before we get on to biologics, can I ask you about combination therapy? You were always quite keen on that, and I know many people were not, because myself included was very, very skeptical that you could combine, as you say, a potential cytotoxic agent with other agents without increasing toxicity. But you were always assured that that was worth doing. And I wonder what was the basis of that?
Well, partly because as good as methotrexate was, we still had a substantial number of patients that still had active disease. And the question is then, if we started combining drugs without a lot of scientific rationale, I'd be the first to tell you that there was not a lot of science in what we tried to do. We took available drugs that we were using in RA that we were comfortable with and started combining them together. And some of the combinations worked and a lot of them didn't. But we developed at least expertise on how to approach the question, realizing that if methotrexate was the anchor drug, we would leverage drugs on top of it and then see what happens. And I think all of us would agree today that almost all of the drugs we use work better with methotrexate than as monotherapy.
Yeah, we got very good at rehearsing the argument that the side effects of the disease were worse than the side effects of the drugs. We were doing so badly, it gave us actually a lot of leeway in experimentation. But the next big change, again with you central to it, was the introduction of biologics. How did you see that developing?
Well, it's interesting. I mentioned earlier that when I talked to you about our first foray into that was looking at interferon gamma, which is one of the first biologics that were looked at. The early history of biologics was not pretty. We all think that, we just jumped into anti-TNF therapy, a lot of it because the work coming out of the UK with Tiny Maney with Tiny Maney and Mark Feldman at the Kennedy. But the 10 years before that was a rough 10 years. Both you and I, and led by Gabriel Panay, did a lot of work looking at depleting the T cell, anti-CD4 therapy, CD5 immunotoxin. I worked with John Isaacs and others on Campath 1H, which was a challenging experience. And all of these biologics ended up in the open studies being positive, but in randomized trials being no better than placebo with potentially a lot of side effects. And it wasn't until directed cytokine therapy moved forward that we got to the drugs that we have today. What a lot of the listeners may not know about is that there were two parallel tracks moving forward with cytokine inhibition. One was directed against interleukin-1, and one was directed against anti-TNF therapy. Both drugs, both concepts were actually being studied in other diseases, primarily septic shock. So with IL-1 inhibition, sepsis was the big draw, but they failed. With anti-TNF therapy, sepsis was the initial draw, and they failed. So drug was available, and investigators then began to explore their use in rheumatoid arthritis. And obviously, anti-TNF therapy was the winner, because even though IL-1 blockade looked great in the animal models, it certainly did not achieve the efficacy that we had hoped.
Yes, that was, I suppose, the turning point was the evidence, perhaps from the ATTRACT study, that showed that halting of radiographic progression was almost universal. As you say, totally against the animal models where we felt that IL-1 was going to be the main mediator of bone disease. But you were involved with one of the earliest publications with etanercept. Were you predicting what you found, or were you surprised by it?
So our first work was with lenercept, the P55 soluble receptor. I was approached by both Roche and Immunex to look at their molecules. I'd been consulting to their companies, and I elected to do my first trial with lenercept because I thought it looked like a better molecule, and I thought Roche had a better concept on how to do drug development, because Immunex was obviously an unknown company with limited research experience in that area. As it ended up, because of manufacturing issues, lenercept became a non-player. Huge amount of blocking antibodies against lenercept due to change in glycosylation of the molecule. And then I moved forward working with etanercept, both in the phase 3 program, but my major interest was combining etanercept with methotrexate, which I had proposed to Immunex years before. And as it was, we did the first combination trial with etanercept and methotrexate. It was designed as a safety study, but the results were so robust that the FDA accepted it for labeling purposes to label etanercept with methotrexate. And, you know, when we did the trial, we had, quote, blinded clinical observers, but you could be blindfolded and still be blind and be unblinded, because the patients who are getting etanercept felt so much better than the patients getting placebo. You'd walk in the room and see the study patients, half of them smiling and half of them miserable. So you found out pretty quickly what they were on.
Yeah, you mentioned that you think methotrexate works with most other anti-rheumatic drugs. The mechanism of that action with the monoclonals, especially the chimeric, it's obvious that reducing antibodies is one, and there's the pharmacodynamic effect as well. How do you think why is it working well with a fusion protein like etanercept, do you think, Mike?
I have no idea, and that's, you know, still an important story. As you mentioned, methotrexate increases the concentration of the monoclonal antibodies by 20 to 25%. So it's a cost-effective way to treat the disease. Rather than increase the dose of the monoclonal, put the patient on background methotrexate. By increasing the concentration, you basically can reduce antibody production. Etanercept, methotrexate does not increase the drug levels of etanercept, and etanercept has a very low level of antibodies. So why it works, we don't know. It's very clear that methotrexate added to anti-TNF increase the efficacy of those compounds. But there are other drugs. Methotrexate increases the efficacy of abemaciclib. It increases numerically the efficacy of the anti-IL-6s, and even with the JAK inhibitors, there's a numeric improvement with methotrexate as compared to the JAK itself. It's not statistical.
So still a lot to learn, I suppose, in that area. But as you say, I suppose the we are particularly concerned, the way we always start everyone with methotrexate, unless it's toxicity, but we get less concerned about withdrawing it after a few years. Do you have the same approach?
Yeah, so number one, I think after, I don't know, 30 some years of methotrexate, approved in 1988, we know everything bad about the molecule. It's actually, most patients do pretty well with it. Tolerability has always been the problem. Nausea, the big reasons why patients stop the drug. You know, respiratory infection rates are pretty low. Hepatotoxicity is pretty low if you monitor patients. My goal is to, most patients want to get on a lower dose of methotrexate because they just don't like taking it. So what I try to do is, as we've published previously to you, is I put patients in clinical remission. And then once they've been at that level for a while, then try to back down on the methotrexate dose. And I tend to go very slowly. I lower by 2.5 milligrams a month because it takes that long for the disease to return. If they're on a monoclonal antibody, I tend to keep them on a dose of methotrexate, generally 10 milligrams a week, so I can reduce the development of antibody production. Then on the back end, if they're doing great with a lower dose of methotrexate and a biologic, I then try to stop the interval of dosing the biologic, which is a cost-effective way to treat the disease. I don't think most patients can tolerate stretching out the dose of biologic without being on a dose of methotrexate because of antibody production.
Yeah, well, I totally agree. We actually have an even longer gap between reducing methotrexate dose, usually 3 months, actually, to get through the half-life of lymphocytes. Mike, you've been involved with small molecules for a long time, and of course, small molecules are suddenly very much part of our armamentarium. Did you think they would become safe enough to use in a widespread fashion, or were you a believer in that they would just be salvage therapy for rheumatoid patients?
Well, I wasn't sure about, we worked first with the P38 MAP kinases. That was going to be the great oral treatment for rheumatoid arthritis, and unfortunately, most of them didn't work. We couldn't get a dose up because of side effects, and when we didn't agree of efficacy, they worked for a couple weeks, and then they had this rebound effect where the CRP went up and the disease flared. So the P38s didn't turn out well. We then started work on JAK inhibitors and obviously and other pathways. I was initially concerned about the JAKs from a safety standpoint, primarily because of some of the early work at very high doses in renal transplant, in which there were a lot of infections, particularly viral disease and cases of Epstein-Barr mediated lymphoproliferative disease. But I think we found that as the dose titrated down, a lot of the serious side effects, particularly the infections, were reduced. And looking back 10 years after these drugs were launched, remarkable with lower doses how we've been able to achieve significant efficacy with much fewer infections. So I'm pretty bullish now about where I think the JAK inhibitors are going to go. I think you mentioned in your podcast the issue of VTEs, which came up kind of out of the blue. We weren't suspecting that this would be a problem. It was identified first in the baricitinib program, and it was also now been reported in the cardiovascular outcome study with tofacitinib, but we're not seeing it so far today with 2 other drugs that are approved, upadacitinib and phlebotomib. And I'm not sure yet what the mechanism behind that is, and ultimately what the risk is going to be with these agents. Taking that out of the side, if that was not kind of in the background, I think the JAKs would be launched much earlier in the disease course, because I think their safety profile is becoming well recognized and manageable. We vaccinate patients in the United States. In fact, we reported several months ago our initial experience with Shingrix in our first several hundred patients with rheumatoid arthritis and lupus at our center. And we reported out that there was not a flare of the underlying rheumatic disease. Vaccination reactions occurred in about 20% of patients. which is what's been reported in the package insert. What we don't know is whether the vaccine works or not, because that was not the purpose of our observation. We really do need some good vaccination studies to tell us whether the new vaccine prevents the development of shingles, because that's obviously the greatest risk factor, the greatest infectious risk factor that we have with the JAKs.
So in an ideal world, when would you vaccinate rheumatoid patients?
Well, I vaccinate everybody now above the age of 50. So I generally start to vaccinate them before they even start methotrexate. Partly because
you'd wait to start methotrexate until they're vaccinated?
No, I'd give them their first shot, start them on methotrexate, and then 3 months later, they'd get their second vaccine. We know that methotrexate may blunt vaccination response, so I would like to get it on board earlier. And in the United States, there's been prior to COVID, there was a huge push to vaccinate patients against shingles. And we had unfortunately, a nationwide shortage of the vaccine, which was a huge problem. But I try to vaccinate everybody early on. So pneumococcal vaccine should be given before they start methotrexate, but I don't wait. I want people to get their rheumatoid treated.
Mike, in your presidential address, when you were president of the ACR, you were talking about the best of times, the worst of times. What were you referring to then?
Well, part of the excitement of rheumatology back in 2001 were the new drugs. We had anti-TNF therapy for a couple years. They had clearly changed the course of rheumatoid arthritis. And I think we were incredibly excited about how we could manage this disease. The worst of times was the high cost of these drugs, which in retrospect, they were low compared to the way they are today. The difficulty with managing complicated rheumatic disease with limited armamentarium, particularly in other diseases besides rheumatoid arthritis. At that point, we had a huge, as we're now seeing again today, workforce problem. We had a major shortage of adult and pediatric rheumatologists. We're now starting to see the same workforce problem again. And then the challenge, at least in the United States, of regulators telling us how we should manage our patients. And that hasn't changed yet either. The insurance companies still basically tell us, very much like in the UK, which drugs we can use and when we can use them.
It's interesting, Michael, we've got almost total penetration of the biosimilars into the market, so that we're not really allowed to start patients on originators unless there's a very, very good reason. And the cost is about a fifth of what we were paying for the originators. The question I'm always asked in the States is, how many more patients can you treat? And the answer is none, because they're exactly the same criteria. So at the moment, it hasn't benefited the patients. It may have benefited the NHS a bit by saving money. But I know it's a difficult question, but why do you think there's such poor penetration in the States?
Well, partly because the only therapies because of patent issues that we can use are biosimilars to infliximab. Even though biosimilars to adalimumab and biosimilars to etanercept have been approved, because of patent law, they're not allowed to be used. So we have biosimilar infliximab, and none of the manufacturers of the biosimilar infliximab really significantly lower the price of their drug. So, it was more hassle to write for the biosimilar than for the originator. So there's been really almost no cost reduction with the biosimilar infliximab, and their penetration reflects that. I mean, at least at our hospital center, primarily we're using the originator. I think in 2023 or 2024, when the patent laws go away for adalimumab, hopefully we'll see some price modification actually, because in the US, the cost of these drugs are now in excess of $70,000 per year retail. That's not the cost that actually occurs, but that's a huge jump. Remember, Paul, when we started When etanercept, the first anti-TNF approved in the United States, was $12,000, and now we're north of 70. So we need some price modification.
It's certainly almost a log factor less here. Well, it is a log factor less, actually, the price. So there are huge differences. Mike, I'm going to finish by just asking you, what do you say to a patient newly presenting with rheumatoid now?
Yeah.
When they see you for the first time and that they ask about their future, what sort of comments do you give them?
Yeah, I'm incredibly optimistic about what we can do for our patients now with RA, Paul. Just like you, my goal is to start someone basically as soon as they walk in the door. You don't need to be a rocket scientist to tell whether someone's got active rheumatoid arthritis. We as rheumatologists train on our expertise in history and physical exams. And whether someone's got synovitis early on, and once you exclude the other causes of inflammatory arthritis, I start them immediately on a disease-modifying therapy. I tell my patients that and I don't even think it's aggressive therapy. I think it's appropriate therapy now. Rapid introduction of methotrexate, wait several months, and if they're not achieving the target goals, adding another drug. Rapid dose escalation of metho, and then 2 to 3 months down the road, if they're not significantly better at doses of 20 to 25 milligrams per week, I'm adding another drug. It's generally a biologic right now, but over time it could very well be, if the VTE issue gets resolved, a JAK-INM or another oral molecule. And I tell them that there's an excellent chance that they'll be able to do everything they want to do, including marathon runners. I mean, I have a number of athletes that I manage, and my goal is obviously to allow them to do all the physical activities that they want to do. And when they ask me, do I think they're going to be able to come off drug therapy? I tell them the goal is to put them into remission and stop the disease. And I don't know whether we're going to be able to stop drug therapy over time, but in many patients, they'll be able to lower the dose to maintain clinical response. So I think of all the diseases we manage as rheumatologists, I'm most gratified when a patient with RA walks in the door, because I know that the vast majority of those patients we can put into low disease activity or remission if we use our drugs appropriately and if patients can tolerate the medicine.
Mike, well, we have come an awfully long way in 40 years, because that's probably the last thing you would have said to a patient 40 years ago. It's been a great pleasure talking to you and reminiscing of the last 40 years of rheumatology. I hope the audience has enjoyed it. If you have, please subscribe to CSF on your favorite podcast app and visit the CSF site to download the latest paper slide summaries. Thank you very much for your time, Michael.
Thank you, Paul. Great to talk with you.